So I have to break up my recorded lecture into less than 15 minutes. That's why I'm making the segments. So anyway, bacteriophages, okay, or unique entities, they infect the host cells, including bacterials. So scientists are wondering, okay, so those viruses, when they inject their genomic material, okay, what genomic material do they inject into their host cell? Because that material, whatever that is, if it is injected, at the end they can make duplicate or the copies of their entities inside of the host cell. So that must be really important substance, right? So Hershey and Chase in 1952 did this T2 bacteriophage blender experiment. Okay, so the question was, what is injected by T2 bacteriophage into the host cell E. coli? Okay, is that maybe DNA, RNA, or proteins? That's what they were trying to ask. Okay, so to carry out the experiment, to answer their question, so they used two different radioactive isotopes. Okay, one is called P32, the phosphate-based radioactive isotope, and the other one is S35, that's a sulfur-based radioactive isotope. There's an important reason for that. So P32, okay, the phosphate is a unique, entity. If you look at nucleotides, DNA, RNA has phosphate in them because they have sugar phosphate backbone. Okay, and the DNA, RNA is made out of sugar, phosphate, and then nitrogenous base, right? On the other side, if we look at protein molecules, okay, proteins are normally made out of 20 amino acids, and there are unique structures of 20 amino acids, but the unique element that is making protein is actually sulfur. Okay, so if you want to distinguish between DNA, the nucleotides, and the proteins, the unique elements that are found from DNA is phosphate, and the unique elements that are only found from protein is S, so sulfur. Okay, that's why they decide to use S35 and P32 to label either proteins or nucleotides selectively. Okay, so when they grow these bacteriophages on the growth medium that is feeding the E. coli cells, but if that growth medium contains S35, radio-labeled sulfur, okay, and then have those bacteriophages inject the E. coli cells, okay, then they can test that if the injected genetic material bacteriophages are proteins or DNAs by tracing what's going into the E. coli cell. How do they trace them? Okay, after incubating both the E. coli cells and the bacteriophage together on this selective medium with the S35, and then they use the blender, okay, and then they disturb the structure by grinding them. Okay, so this structure is disturbed by this blender. So now they are broken into pieces. So these bacteriophages outside and the cells were separated after blender, and then they did a centrifuge. Centrifuge is what? It's like your washer machine. It separates things based on their weight difference, right? So when they did that, so the liquid on the top and the heavy stuff at the bottom, we call the pellet. Okay, so the things are separated based on their weight, right? So if you have a mixture of these guys, okay, which one will be considered as a heavy stuff? Probably it's these guys. So the bacterial cells will be coming down as a pellet at the bottom. On the other side, these things will be still floating because they are so light on the top as a supernatant, right? So they try to measure where the radioactive isotope is. Okay, the S35 in this case, is it coming from bacteriophages still floating in the supernatant, or is it in there inside of a cell because they are injected by the bacteriophages? So they traced it using a Geiger counter. Okay, so when they did that, they found that S35 was still in this liquid supernatant. That means they were never injected into the host cells. When they repeat the same experiment, okay, now when bacteriophage and E. coli cells are grown on the medium that contains P32. Okay, so P32 is incorporated into DNA RNA molecules, right? And they incubated and then did a blender, okay, and then they did a centrifuge. Now they try to trace where the P32, the radioactivity is coming from. Okay, is it in the supernatant or at the pellet? If they found that the radioactivity is coming from the pellet where the bacterial cells are. So what does it tell you? Okay, so it must be either DNA RNA that is using this P32, okay, and then that DNA RNA is actually injected into the bacterial cells. Okay, but the whatever is injected is not using S35 because that's never injected into bacterial cells. Okay, so what does that tell you then? So most likely bacteriophage has genetic material that's made out of either DNA or RNA. That was the conclusion. So this conclusion combined with the previous one done by Avery et al. And the Griffith et al. Both are telling what? It's either DNA or RNA molecules are used as genetic material in maybe bacteriophages as well as what? The bacterial cells, the pneumonia, Micrococcus pneumonia cells, right? So those are the two major findings suggesting that it's a nucleotide, either DNA or RNA. Those are the genetic material, most likely DNA. Okay, so back in 1950s, 40s and 50s, scientists were very excited by those findings. So a lot of people worked on DNA molecules to identify what their structure look like and what they are made out of. Okay, there are a group of scientists, especially in England at the time, doing a lot of active research. So Rosalind Franklin, and who was working as a postdoc at Maurice Wilkins' lab in Great Britain, okay, they did a novel study on the DNA structure. They used X-ray crystallography, which measures the diffracted pattern of X-ray beams after crystallizing DNA structure. They shine the X-ray beam to the DNA and then collect the diffracted beams of X-ray, okay, energy. And by using that, you can identify the structure of the DNA. That's what X-ray diffraction pattern is. Okay, so Rosalind Franklin did a lot of experiment, hands-on experiment, and she gathered the data. And this is a picture of her data showing DNA has this kind of structure. Okay, so kind of unique structure. What does it look like? Maybe some kind of hole, and in the middle of the hole, some things are going through there. Okay, so that is a structure based on her scientific data. Okay, at the same college in England at the time, there were a couple of scientists, okay, who were working on the project in a different lab. Okay, so those guys. Actually Watson and Crick, but before we get to Watson and Crick, there's another group of scientists, Shagap et al. Okay, they studied on the structure of the DNA as well. 1952, they isolated DNA molecules from different living systems. Okay, and they tried to understand the makeup of the DNA, and they understood DNA molecules are made up with different nucleotides. Okay, there are 4 different nucleotides: adenine, guanine, thymine, and cytosine, 4 different kinds. And they wanted to know if the makeup of those nucleotides in DNA molecules, if there's some kind of rule or ratio in the DNA molecules that are isolated from different living systems. Okay, they isolate DNA from different living systems and try to look at some kind of pattern, on any conjugate pattern. And there was some kind of conjugate pattern. What they found was, regardless of where DNA was isolated, okay, the total amount of purine bases, purines are A and adenine and guanine, okay, that is same as the total amount of pyrimidine bases, which are made out of thymine and cytosine. Okay, so purines are doubling structure, okay, nucleotides, and pyrimidines are singling structure. Okay, somehow the amount of purine is same as pyrimidines. That was another important finding, giving some hint about the DNA structure. Okay, so based on Rosalind and Shargap's group's discovery, a couple of scientists back then in 1950s who were studying at the same college in England, okay, they came up with this structure of the DNA molecule. Actually, James Watson was an American, but Francis Crick was a British scientist. They worked together. Okay, so especially Franklin, Rosalind Franklin's data was very important in illustrating this structure. If you look at this double helical structure that we are familiar with as a DNA structure, from the top, especially if you cut it up, and if you look at from the top view, you will see that circling structure of DNA, and especially that phosphate sugar backbone is going down as a spiral. Okay, so that's what Rosalind saw exactly, and these two scientists came up with this three-dimensional model of DNA based on her, I guess, data, as well as the purine and pyrimidine, the ratio. Okay, if you have adenine and guanine, those are the purines, matched up with thymine and cytosine, the same rate, then you'll have, that equal ratio. A plus G should be same as T plus C because they are paired up, opposite in the DNA structure. So if there are two strands going this way, and each strand has the base paired up with the other strand's base, okay, A and T and G and C, yes, indeed, you will have the double-strand DNA structure. Okay, so the James Watson and Francis Crick got a Nobel Prize by identifying this three-dimensional DNA structure back in 1953. And one of them is an American, actually graduate from Indiana University. Actually, James Watson was IU graduate. Okay, he graduated from, I think, 1950, I think. Okay, so that was one of the major discoveries of molecular biology in the previous, I guess, century, 20th century. Okay, very exciting. And then other scientific data were also made available, especially Arthur Kornberg discovered the enzyme that can duplicate, they can make copies of DNA. He made RNA polymerase enzyme discovery back in, I think, 60s. Okay, so he used in vitro system that can replicate DNA molecules, especially using E. coli as a model system, and they grew up E. coli cells and lysa cells and prepared extracts out of the E. coli cells, and then he separated out different extract molecules from there, and he identified an enzyme by doing so. That was a DNA polymerase enzyme, and he identified that that enzyme can make copies of DNA, so he named it as a DNA polymerase number 1. Okay, so the first scientist who discovered the enzyme that can duplicate DNA. And then another major discovery was done by Messelson and Stahl back in 1953. Okay, they identified when DNA molecules are replicated, okay, the replication has a certain pattern. The pattern is, actually, we know that it's a semi-conjugative pattern. So what does semi-conjugative pattern means? When an original DNA is copied, okay, then it's made into two DNA molecules, so single molecules becoming two, right? So when you do that, how is the original molecule used? Is the original molecule left over as intact and you make a new daughter strand, or is the original molecule used somehow to build the daughter molecules? What is the secret? Okay, it turns out the secret is one strand of the original molecule is given to the daughter strand. Okay, on another strand of the original molecule is given to the daughter strand. So that's the semi-conjugative replication. So how did that discover? So this is the experimental setup. Okay, he used another radioactive isotope N15 in this case, and then switched the medium containing N15. So 14 and 15. Okay, so originally the entire DNA will be labeled with N15 first. Okay, and then when the replication happens, okay, they will switch out to N14, right? But when they try to see the newly made copies, how they are labeled with those radioactive isotope, they saw that all the newly made DNA has that uniform there. Okay, and then when they trace that continuously, is going there. So the uniform DNA is split into another half and going continuously. So this pattern explains, yes, indeed, it's a semi-conservative way how